Hi students, today we are going to discuss about important 5-mark question, lipoproteins. Your university question will be like this: classify lipoproteins with their functions. Okay, so you all know about lipoprotein means the composition are lipids and proteins. The introduction point for this lipoprotein is usually lipids. The definition of lipid is it is insoluble in water and soluble in ABC, that is alcohol, benzene and chloroform. So how these lipoproteins are transported? Lipids are transported with the help of lipoproteins only. The lipids are, that is cholesterol and triacylglycerols, transported from one place to various places, right? So transport of cholesterol and TAG like lipids to various site is the function of lipoproteins. Okay, coming to the structure, just one or two points only. You have to draw the diagram of lipoproteins for your 5-mark question. Actually, in the center core, it is occupied by triglycerides and cholesterol ester, which is surrounded by a layer of phospholipids and cholesterol, and finally one peripheral layer that is made up of apoproteins. These three compounds are the important points for your structure of lipoprotein, plus you have to draw the structure. Next one is, how will you separate these lipoproteins? Here the technique is electrophoresis. Depending upon the size and density, the lipoproteins are separated. So this is three types of lipoproteins are in the electrophoretic field depending upon the size and density. One is alpha lipoprotein, another one is beta lipoprotein, third one is pre-beta lipoprotein. All three are very, very important MCQ points. That is why I am giving in detail about the electrophoresis of lipoproteins. Next, coming to the classification, we have 4 classification of lipoproteins, that is chylomicron, VLDL, LDL and HDL. We will see one by one. First one is chylomicron, which is derived from intestine, and the main composition is triacylglycerol. Here the apolipoproteins for chylomicrons are mainly B48 and also apolipoprotein A, apolipoprotein B and apolipoprotein C. This is also another important MCQ. The function of these chylomicrons are, it transports exogenous fats, that is dietary lipids, to various tissues. This is the function of chylomicron. And coming to the degradation of triglycerol, which is present in the chylomicron, is by the lipoprotein lipase, the TG, that is triglyceride present in the chylomicron, is converted into fatty acids and glycerol. This is the degradation point for triglycerides which is present in chylomicron. Okay. The second lipoprotein is VLDL. For this VLDL we are having only 2 or 3 points. It is otherwise known as pre-beta lipoprotein. This VLDL is synthesized from both liver and intestine. The function of VLDL is it transports the endogenous triacylglycerols. Chylomicron function is transports the exogenous fats. Here VLDL transports the endogenous triacylglycerols, right? And the next one is LDL, that is low density. VLDL means very low density lipoprotein. LDL means low density lipoprotein. It is otherwise known as beta lipoprotein. This is actually bad cholesterol because the function of LDL is it transports the cholesterol from the liver to peripheries. So from the liver, all the cholesterol is distributed to the peripheries where it will get accumulated or deposited in the peripheral tissues. That will lead the cause for atherosclerosis, that leads to myocardial infarction. That is why LDL is known as bad cholesterol. It's synthesized in liver from VLDL, and the composition mainly composed of cholesterol ester. The main apolipoprotein is B100. Chylomicron, the main apolipoprotein is B48, whereas LDL, the main apolipoprotein is B100. In biochemistry, First topic, biomembrane transport, where you have read about the endocytosis and exocytosis. In LDL metabolism, the mechanism is receptor mediated endocytosis. Suppose they are asking in MCQ or sometimes Viva, they will be asking receptor mediated endocytosis occurs in which metabolism, means LDL metabolism, right? Next lipoprotein is high density lipoprotein, otherwise known as alpha lipoprotein. This is called as good cholesterol because it transports the cholesterol from peripheries to the liver. From the peripheral areas, this HDL, that is high density lipoprotein, it scavenges all the cholesterol from the peripheries and transport it to the liver. From the liver through bile duct and bile, it will get excreted. That is why HDL is known as good cholesterol. It is this mechanism is known as reverse cholesterol transport. This reverse cholesterol transport by HDL lipoprotein, that is high density lipoprotein, and the enzyme is LCAT, lecithin cholesterol acyl transferase. Here the HDL, the synthesis is from liver and intestine. Major apolipoprotein is lipoprotein A, apolipoprotein C and apolipoprotein E. Main composition of HDL is phospholipids and cholesterol, right? And the last one is, this is very rarely asking MCQ, free fatty acid is always bound with albumin. So the transportation for the free fatty acid is by albumin. Next important Viva question or sometimes MCQ question is, name the enzymes involved in lipoprotein metabolism, right? Very rarely some examiners will ask about the enzymes involved in lipoprotein metabolism. Very simple, LCAT, lipoprotein lipase and hepatic lipase. Simply 2 lipases and 1 acyl transferase, that is lecithin cholesterol acyl transferase, lipoprotein lipase, hepatic lipase.